Okay, so in this screencast, we're going to talk about pathogens and infectious disease. I would think that we could anticipate this one to show up on the AP test in May, just given the um, the fact that we're right now currently in the middle of a pandemic. So let's learn about it. It's pretty important stuff, obviously. Okay, so the term pathogen refers to any organism that can cause disease, and there are basically four types of these pathogens. There are viruses, which we're dealing with right now with the coronavirus. There's bacteria, which cause a lot of human disease. There's parasites, which include things like worms, amoebas, and things like that. And then there's fungus, which can also cause human disease. So pathogens get passed from host to host in different ways. So the main ways they get transmitted would be body fluids, so droplets from coughing and sneezing. That's what's going on with COVID-19. Sexual fluids, that there's a lot of sexually transmitted diseases like HIV, herpes, HPV. Uh, a lot of them are transmitted through contaminated water. So if you drink water that has certain things in it, like cholera bacteria or hep A bacteria or the Giardia organism, you get sick. And then there's animal vectors. So that means that in order for the organism to go from one person to another person, we'll just focus on human transmission, you have to have some sort of an in-between emissary. So like a mosquito, a flea, or a tick that will take it out of one person and put it into the other person. All right. Now, so pathogens that are transmitted from drinking water would include things like cholera, amoebic dysentery, Giardia, guinea worm. And the things about these is that there's a social justice component to this because in countries that are developed where they have ways of purifying water before they pump it into people's houses, people do not suffer from these diseases. These diseases are definitely uh, a burden on the poorer countries of the world, the developing countries of the world that don't have the infrastructure to provide clean drinking water to their citizens. As I said before, some pathogens require a disease vector. So a really important example of this would be malaria. My father had malaria, and the thing is, once you get this, you pretty much have it your whole life. It's basically a protozoan that can live in your liver and your bloodstream. And so what happens is a person gets infected. It takes up residence in your liver. It starts going around in your blood. A mosquito drinks your blood. That mosquito goes and bites another person, and a little bit of that gets into this person, and then it gets transmitted around that way. So, uh, as I said, it's very difficult to get rid of some of these organisms once they're inside a person's body. Sometimes they'll respond to medications, but sometimes, like with malaria, they often don't. And so, the best way of preventing them is to try to eradicate the disease vector. So, for instance, DDT we've established is a really bad thing, but there's still countries that use it, and the World Health Organization permits it because malaria is such a devastating disease; it kills a lot of people, and the best way of stopping its spread is to stop the mosquitoes that carry it. Now, most pathogens can be killed by the use of antibiotic drugs. So these are chemicals that we put into our bodies. They poison the pathogenic organism without killing us. They might harm us a little bit, but the idea is they're going to basically be poisonous to that organism that's inside our body. And antibiotics, for the most part, work really well against bacterial and fungal infections. So if you go to the doctor and you've got a bacterial like bacterial pneumonia, I had it one time. They gave me these pills. You take these pills, you're done. If you get like a rash in your body and it's some sort of a fungal infection, like ringworm, you just spread this stuff on, this cream on here, and it gets rid of it. It kills it. You're poisoning it, right? Now, the thing is, parasitic infections, sometimes they can successfully be treated by antibiotics, but a lot of times they're hard to get rid of because you had to know exactly which one it is, and that can be hard to diagnose. And sometimes there's just nothing you can do to get rid of it. Um, and the other thing to know is that antibiotics don't work at all on viral infections like coronavirus. So you can't just take a medicine to make the virus go away. And the reason you can't do that is because antibiotics work by poisoning things. And you can't poison something that's not alive. And it turns out that viruses are really kind of this, they're not really alive. They don't meet most of the criteria of being alive. And so they can't be killed with poison. So we have to get rid of them in a different way. But before we talk about how we get rid of viruses, let's just talk about one important thing about antibiotics, because this is definitely something that is a concern. We talked about this when we talked about concentrated animal feed operations, where they just give antibiotics generally to the organisms. But the idea is this, that you're supposed to, when you get diagnosed with a bacterial infection, the doctor says, okay, you need to take these pills. You need to take all of them for say 10 days. Take them twice a day for 10 days until the bottle's gone. Now, right now, you're feeling terrible. Well, the idea is you take the pills and it starts killing them. Well, it turns out there's genetic variants, right? There's genetic biodiversity in this bacterial population, and some of them just by random mutation chance happen to be resistant to that particular medicine. And so what happens is when you take it, the ones that don't have that gene for resistance, they die much quicker. The ones that have the gene for resistance are harder to kill, so they linger a little bit longer. At this point, though, several days into your treatment, you're feeling better. But you better keep taking those pills because you still have some of them in you, not enough to make you feel bad, but they're still there. If you continue to take the medicine, you'll kill all of them. And now there's nothing left to multiply. This is how it's supposed to work. There's none left to pass on that resistant gene. But the problem is this. Often what happens is, again, I'm sick. I have all these bacteria in me. Some of them just by random genetic chance happen to have this mutation that makes them resistant to this particular antibiotic. I take the pills. I kill most of them. The ones that die quickest are the ones that don't have this genes. The ones that are resistant are less likely to die. At this point here, I'm feeling better. I don't feel sick anymore. And I go, you know what? I'm not going to take these pills anymore. Well, the problem is, since I didn't kill the entire population, the ones I didn't kill, they multiply. And after a while, they become the dominant members of the population at large. And so now you have these uh, pathogens that are resistant to antibiotics. And now you've got to find a new antibiotic to kill them. And you may not be able to find that. So there's definitely many bacteria out there that are resistant to most of our antibiotics. It's actually quite a crisis we're facing because a lot of the drug manufacturing companies don't want to invest the money to make new antibiotics. And the ones we have are becoming of limited use against many of these pathogens that have developed resistance in this way. Okay, now let's talk about viruses. All right, obviously, that's a big deal right now. Okay, so antibiotics don't work against a virus, as I said, because a virus isn't really a living thing. A virus is basically a protein capsule that inside it has this either it has nucleic acid, it has RNA or DNA. So COVID is an RNA virus, but there's also DNA viruses out there. There's a lot we could say about viruses, but let's just consider this. It's essentially this little protein capsule that has DNA in it. Now, normally a cell replicates its own DNA, but this doesn't have any ribosomes. It has no mechanism for replicating. It just doesn't have things that are going to allow it to make more copies of itself. So what happens is it basically has these proteins on its surface, and these proteins will match the proteins in a host cell. So the host cell is tricked to allowing it to put its genetic material inside the host cell. So basically, the surface proteins will match a protein on the surface of a cell membrane, and then when that happens, the virus is able to inject its nucleic acid, either DNA or RNA, inside the cell. And once that happens, then it tricks the cell into making copies of itself. So once it gets inside, okay, so in this case, this one actually entered the cell itself, it broke apart. Now out comes the viral RNA. Now that RNA starts interacting with our ribosomes. In some cases, it goes into the nucleus of the cell and gets transcribed here by the mechanism that replicates our DNA. But the bottom line is the viral DNA, the viral RNA or DNA is being replicated by our own cells, by the ribosomes in our cells are going to assemble the new protein coatings, and then once our own cells have built the new virus, we release them. So the thing is, you can't kill this because it's not alive, it's not eating, it doesn't take anything into its body, so you can't kill it. And once it gets inside your body, in your cells, it makes copies of itself. So the trick is somehow we need to get rid of these things without poisoning them. Well, it turns out our body has a way of doing this. So what we do is instead of trying to poison them with antibiotic drugs, we have these in our blood system. We have immune cells, white blood cells, that are designed to identify and destroy invading pathogens. Those can be bacteria, viruses, and other things, but we'll mostly focus on bacteria and viruses. So your bone marrow produces your blood, and it produces, among other things, these things called white blood cells. There's lots of different white blood cells. I want to focus on one in particular called a B cell. So what a B cell is, it's a white blood cell that grows these proteins on its surface. And these proteins are called antibodies, which I'm sure you've heard of. So an antibody is just a protein that grows on the outside of these B cells. Now, on these little Y-shaped proteins called antibodies have these surfaces at the very ends of them, the termination of these things, have different shapes to them. And the idea is the antigen, which is basically the pathogen, it has proteins. And if the shape of a protein on a virus, for instance, matches the shape of this protein on the end of this antibody, then the two click together and the antibody sticks to the virus. That's the idea. And so what happens is in your bone marrow, you just generate these randomly shaped proteins on the ends of antibodies. So every moment of your life, you're making hundreds of thousands, millions of these cells. Each one of these B cells is going to have just a randomly generated um, protein structure that may or may not match anything in that of a pathogen. And so they course through your body, they live a few days, and they die, and your body's just constantly making new B cells, each of which has a brand newly newly minted protein here. Okay. Now what happens is, if that new shape that you made in your B cells in the proteins happens to match the surface protein of a virus or a bacteria, then the antibody will stick to it and will basically get pulled off of this B cell. Now it's a little bit tricky. Another cell has to come along and tell the B cell that this has happened. Okay, but once it has been activated, once it has made connection with uh, a virus or any pathogen that's gotten into your body, once the antibodies begin to stick to it, then what happens is this B cell knows that, hey, this randomly generated protein shape that I made for these antibodies is an important one. And so what'll happen is your body will begin to replicate these things like crazy, and they'll start to swell up in your lymph glands. So you'll get, you go to the doctor, they go like this, right? That's because this is where you generate these B cells, okay? And they multiply and they multiply and they multiply, and they just start blasting out antibodies. One of these B cells will just blast out thousands and thousands and thousands of them. And you've got millions and billions of these cells coursing your body just pumping out antibodies. And the idea is, I want to stick antibodies onto viral particles. Why? Well, the reason is, once the antibodies that get pumped out by these activated B cells make contact with the viruses, they stick to it. And what happens is you have these other white blood cells called macrophages, and they go around, and what they have proteins, and these proteins are designed to match the other end of the antibody. And when this clicks here, then this white blood cell just engulfs this one like an amoeba, and it just destroys this. It just breaks it apart. And so it's a beautiful mechanism. The idea is, as long as you can mark with antibodies the virus faster than it can replicate, then you win the battle. If it replicates faster than you can identify it and destroy it, then you lose the battle. All right. Now, what happens though is really cool is this: once your body has encountered this and you've activated a particular B cell that makes a certain kind of antibody, say for chickenpox, okay? You got exposed to chickenpox. Now your body knows, like, hey, this is the antibody for chickenpox. Well, what it does then is, in addition to making these what are called plasma B cells that pump out all these antibodies, it will also make a separate copy that's called a memory cell. And these memory cells won't pump out their um, antibodies, but they'll just keep replicating in your body. They'll always be in your body. There'll be a population floating in your body, potentially for the rest of your life, because your body goes like, hey, this particular antibody, it matches something that's out there. And so, like, that you get exposed to, say, chickenpox when you're 10, and this happens in your body. Well, you have these memory cells that just linger in your body, and now you're 40 and you get exposed to chickenpox again. You're already ready to go. You don't have to wait for some randomly generated B cell to have this, randomly generated antibody. Your antibodies are ready to go, and as soon as they get activated, the process starts over again. But it's able to jumpstart and get ahead of the virus very early on, so you never really feel the infection. So that's how our immune system gets rid of viruses. It's this combination of B cells making antibodies and macrophages devouring any viral particles or bacterial particles that happen to have these antibodies stuck to it. Now, here's how vaccines work, okay? So for a vaccine, what I want to do is I want to make a surface protein for whatever virus I'm concerned about. So, or bacteria. So I don't want to, I could either take a whole virus and somehow neutralize it, get rid of its nucleic acid or disable it. That's one way of doing it. Uh, what's called live virus vaccine. Or you can just take a little surface protein. And then what you do is you inject the person with these surface proteins, which will bind to the um, B cells in your body. And then you will activate these B cells. Even though I didn't actually give you the virus, I gave you the protein fragment that activated the process. And then you start making antibodies. And so that's why after you get a flu vaccine, or if you get your COVID vaccine, you're going to feel sore for a few days. You feel sick for a day or two. It's because your body is like, I got to make antibodies. I got to make more of this one particular type of B cell. But then you'll make those memory B cells. So if you get exposed to it in the future, you win the battle before you even know that you encountered the pathogen. Now, one thing that's kind of cool about the Pfizer vaccine and the Moderna vaccines that are out right now is instead of injecting you with protein fragments, they actually inject you. It's a brand new technology. They inject you with RNA. And so what happens is this messenger RNA is injected into your body. It gets into the ribosomes of your cells, and it just makes, instead of making the entire virus, it just makes the surface protein of the virus. So basically, it's doing what the virus does, but just the part that lets your antibodies work. It doesn't make the nucleic acid for the virus. It just, this little snippet of RNA is going to make your ribosomes produce the surface protein that's going to activate your B cells. So you'll make these memory B cells. So you're ready to go if you ever encounter that virus for real. Now, one of the things that we should be concerned about right now, and you're hearing about it, are these variants of COVID-19, and that's to be expected, right? So every time DNA or RNA replicates, there is a chance that there's going to be mutations. They just happen, and these mutations sometimes express themselves as in different ways, and right now the ones that are happening, they altered other aspects of the virus's behavior, but they haven't really changed its surface protein. So the vaccines we have right now are still working against the new variants, like the Brazil variant and the UK variant and the South Africa variant. You should expect this to happen. All DNA and RNA, as it replicates, has a chance of mutating. So the more the virus is out there in the world, the more chance there is for mutations to occur. That's why it's important that we don't just vaccinate people in our country. We need to vaccinate the whole world, because there's this huge reservoir of viral RNA right now that's just replicating. And every time it replicates, there's a chance of it making a mutation. And if that mutation changes the shape of the surface protein enough, then our vaccines won't work. And so it's a race against the clock. We've got to get this population of virus down so there's less chance of a mutation occurring that changes its surface protein. Now, how do these novel viruses come about in the first place? Where do these pathogens come from? So we often talk about a reservoir. So basically, in nature, there are organisms that are infected with a particular kind of virus, let's say, or bacteria, all right? And it doesn't have a surface protein that will allow it to infect humans. So it just can't gain access to our cells. So it's out there. There's so many different things out there that have nothing to do with humans. But what will happen is a mutation will eventually occur. And when that mutation occurs, the surface protein changes enough that it can now infect humans. So the deal is, if humans are coming into contact with animals, the more contact there is between humans and animals, especially living animals, the greater the likelihood is that a mutation of a virus or bacteria that's not a human pathogen the mutation will alter it, so it will become a human pathogen. Now, if that happens in a bat and the person never encounters the bat, well, no harm, no foul, right? So there was this mutated, virus in that one bat, or even a bunch of bats, but it didn't get into people, so it never infected people. But when people come into contact with it, that's a problem, and that's why the concentrated animal feed operations are an issue. So when you have things like avian flu or the swine flu, the reason these are farm animals is because or bovine flu. So basically, these animals had the virus in them. The virus mutated. The people were close to the animals. The animals were close to each other. So there was a large population of virus. So it's just like we're saying with the world today with COVID. When you have a concentrated feedlot, you've got and you get an infection. You have a lot of organisms that are infected, and that means there's a lot of replication of virus, and there's a lot of chance of making a mutation. And if this mutation changes the surface protein so that it can infect human cells, humans happen to be going into these feedlots. So this is a real concern for the future. This is how novel viruses get. Now, we don't really know exactly what the initial reservoir of COVID-19. They think it was bats, and they think it had to do with a food market in Wuhan, China. And it makes sense that is how these things happen. Okay, and this is what I just talked about here. So I guess I wasn't looking at my slide. Okay, so now what we'll find sometimes is that these reservoirs are out there. A mutation occurs, it gets into humans, it starts infecting humans. This happens like with Ebola, all right? And then we fight this successfully, we fight it off, and we socially isolate people, and we get the virus out of the human population, but it's still existing in these reservoirs in nature. And so it tends to happen again. So we find, especially with tropical diseases, that there's these reservoirs in nature where these animals have this virus. We get it out of the human population, but it keeps reinfecting the human population. Now, it's one of the things that you're expected to know for this course. I think it is worth noting is that just as the tropical areas have a lot of biodiversity in terms of plant species and animal species, it shouldn't be surprising they also have a lot of biodiversity in terms of pathogenic species. And that's just because there's just more life in the tropics, so there's just more variations of that life. And that means there's more things that can affect people. So tropical diseases, that's why, unfortunately, a lot of the people who get infected with these diseases are impoverished people in this part of the world. Unfortunately, a lot of the developed world is not in tropical places, and they're not exposed to these pathogens. And so a lot of times they don't focus on them that much. But what's happening is like if you travel to a tropical area, a lot of times you got to get, you might get malaria, you got to get this shot, you might get yellow fever, you got to get that shot. Oh, we got to worry about dengue fever, whatever. It's because there's a lot of biodiversity in the pathogenic populations in tropical areas. But what we're finding is that over time, because of global warming, that the zone in which these tropical pathogens can exist is expanding northward and southward from the equator. So we're finding that areas that did not used to have, like malaria, dengue, Zika, are starting to get them because the zone of habitation is expanding as the world gets warmer. So sorry this was such a long one. It's pretty important and it's pretty cool. Hopefully you're able to follow along. I know it was a lot to try to digest. Let me see how I turn this off. I don't like this new way of doing it, but it's what I've got.